first of all, I want to thank the organization committee to invite me to the speech. I'm very pleased to be here and really privileged to be here. And so I've been doing oncology for the last, well, I would say more than 15 years. And my passion is really to bring innovative therapies to patients. So when I think about cancer vaccine, I get very excited about because there are so many different drugs treating oncology patients. But I think really what makes a difference is that potentially the immune system, because you hear people who have cancer, who don't have cancer, the reason could be genetics, could be viral, could be environmental issues, but down to the bottom is the patient's immune system. Why is it some people who can overcome the budding cancer cells and kill it, and some people would have the cancer cell develop into full-blown cancer? Now, the thing is that cancer does not kill until it becomes overt. So my talk today will be talking about the cancer immunology, the cancer vaccine, the pros and cons. I'll talk about different approaches. And then what's the cancer in development? I'll talk about the approaches and some of the approaches that have been used and were not used. And then we'll also talk about a case study that we had Roche, we had a therapeutic vaccine task force that we evaluated all the therapeutic vaccines. So we're not talking about prophylactic vaccines, we're talking about therapeutic vaccines. And therapeutic vaccines are actually treating patients, not preventing disease. And then I also talk about the recent development in the cancer vaccine field, and then also talk about a little bit about the FDA guidance, the draft guidance that FDA has helps with the cancer immunology consortia to help to put together a guidance that may provide industry with some idea as for the clinical evaluation or consideration for cancer vaccine development. And then I will lastly talk about the criteria at Roche that we evaluate cancer opportunities in terms of vaccines. So in terms of cancer immunology, you're really talking about two compartments of I would say immune responses. One would be the immune response from the innate response. So that's what you see the slide, that where you have the B cells, releasing the cytokines and the macrophages, the monocytes, and basically really the inflammatory process which lead to potential developing of cancer. Then you have this other system called the adaptive response, where it's really T cell mediated, and that's where you have your ADCC, CDC, and the T lymphocytes that will kill the cancer cells. So it's really the balance between the innate and adaptive responses, that really determine whether or not the cancer cells will be killed at the get-go before it becomes overt into full-blown cancer. So in terms of the basic approaches in vaccines, I know many of you are experts than I mean, but I'm just kind of trying to look into some of the potential approaches that's being used in cancer vaccines. One will be the peptide. So what I'm looking at, if you see the diagram on the right side, is that you will have an antigen epitope, and you have a carrier, and then you have an adjuvant. So that's typically the sort of the structure or the construct of cancer vaccines, and then we have the peptide-based, the cell-based, the nucleic acid-based, and the viral vector and adjuvant. So quickly that we talk about, this is some of the evaluation we have done internally and looking at the different approaches. So for the recombinant approach, we think that it's definitely the R&D and in terms of the scientific effect, and then going to, it's really injection of purified protein peptide in a soluble vehicle or adjuvant. So going to the pros and cons more deeply, that you'll see that the peptide, we believe that there's a lot of expertise in this area, and what you're talking about is a cancer-specific peptide, a specific sort of antigen that you can actually pause onto the carrier. So then what you do is that you develop a very specific sort of immune response toward that, the antigen. So the question here is that different tumor type may have different antigens. So antigens overexpressed by lung cancer doesn't mean that it's overexpressed by breast cancer, for example. So when you think about cancer, it's really a group of diseases. It's not one single disease. So you really have different expressions of antigens for different tumor types that you have to deal with. Now, another approach that is a cell-based approach. So you have the autologous vaccine. You also have the allogeneic vaccine. For example, Provenge is autologous vaccine. So autologous vaccine is basically you get the dendritic cells, you load it with antigen, and then you refuse back to the patient. And what's nice about autologous vaccine is that it really utilizes the patient's own dendritic cells, which is the best way to present antigens. The cons will be that, it's really a process that you have to do, and then which is kind of cumbersome, and then also that, in terms of how you administer the vaccines, there may be certain centers can administer the vaccines, not typically in the oncologist clinic they can do that. So here's some of the pros and cons that it is very effective, I believe, because the patient's own dendritic cell is the best way to present the antigens. There are many variety antigens that can be presented, not single antigen, so that's good. But then of course the process is really cumbersome. The nucleic acid, meaning that it's injection of the DNA plasmid, which would induce a native immune response. Or you inject a RNA that would induce potentially blocking the translation process and thereby blocking the production of certain proteins. So for the pros that you're talking about, this is really a sort of a variety of antigen that you're talking about. It's not a single antigen. And it's really kind of non-specific in terms of stimulation of immune response. But some of the cons are that, there's certain, so far it's been limited clinical data about it. It's working in fish and then maybe large animal like horses. But in humans, it has been limited in terms of the data. So again, it's really kind of unknown, and also unknown regulatory risk being having a gene therapy. So now also about the vector approach. So the vector approach is that you have a virus expressing immunomodulatory antigens, and thereby, you can elicit an immune response. And when you think about a vector approach, that can be different viruses like adenovirus. So you really talk about a combination of multiple antigens. Again, this is a multiple antigens, not a single antigen. But some of the issue is that, you're injecting a virus. So sometimes the viral antigen may overcome the human antigen, and there may be some safety issue. But I believe that Oncovax had come out with a melanoma vaccine, which is oncolytic virus. It's in phase 3 trial. So, we'll see what's going on, but then, it's an interesting approach. Now, adjuvants is something that you can add to many of the different approaches, and by increasing it's like an immune stimulant. So you can actually sort of magnify the immune response. I mean, a lot of times it's being added to the peptides, by increasing the immune response, because you're talking about one single antigen, and sometimes the immunogenicity could be limited on one single antigen. So what you do want to do is add some adjuvant to stimulate. So a lot of times you see adjuvants added to that. But then again, so here's the pros, is that really ability to enhance immune system. But then here, the problem with that is that, it's a variation. It's the mechanism of action is variable. And whether or not you're going to add to, the peptide or the autologous is not exactly clear. And also the potential, variable side effects that we don't know. So it's also a really good approach that can be used in many, many different approaches. So in terms of looking at all those approaches, you say, well, what is exactly, pharma would be looking at? So just from, the experience that I have evaluating different vaccines with my evaluation team, we tend to like to have the recombinant approach because it's simpler. You have one antigen, and then that antigen, whether or not expressed or not expressed in certain tumor types. You can monitor the effect, monitor the T cell CD8 immune response based on the specific antigen. So you can monitor it where the cell-based and the other, like the cell-based autologous, would not be a business model for us because, you're talking about reinfusing it through process and allogeneic, although you can probably get off the shelf, but then again, you're talking about the actual cells, and sometimes there's issues with the content uniformity. The FDA seems to have some issues with that. So the approach that Roche would prefer to see in general is the recombinant approach. So here's listed, it's not a complete list, I would say, but what I want to show is that really you see all the listing of the vaccines in late stage development. You see that the antigen specific is probably the majority of that, and you see some cell-based and polyvalent. And of course, the recent approval last year in Provenge for prostate cancer, it's an autologous vaccine. And also, I would say it's not really a cancer vaccine, but the ipilimumab, which is a immunotherapy that's just approved for melanoma, which is a very good immunotherapy. So we're very excited about that. There's immunology seems to be now really hot about, everything sort of come out, it's mature, and it's ripening for making a difference in patients' life for oncology. So the case study I want to present is that Roche, we did a therapeutic vaccine task force where we evaluated a couple years ago of all the therapeutic vaccines and in the 5 therapeutic areas listed on the left. And we looked at all the approaches that I talked about. And then also we looked at landscape, and we also had KOL expert, 10 of them. We had a 2-day meeting in Paris, have an off-site meeting, and we evaluate basically a lot of opportunities, and then we then present to management. So the criteria we looked at is that in terms of biological rationale, which I'll go over a little bit more detail later, is that it's really understand the target. Whether or not there's polymorphism of the target, is it variable, is it variation? And also really important is the mechanism, which I'll go over a little bit more detail. And the preclinical pharmacology is important, is looking at the efficacy, what is the good animal model, and really important is looking at the biomarker. We want to make sure that the preclinical efficacy and safety is driven by the target of interest. And looking at the therapeutic index, making sure it's safe, and then very importantly also is the differentiation from the standard of care therapies and the technical effort required, and whether or not we would need to have special expertise. And the clinical data, it's self-explanatory, is that clinical efficacy, biomarker is really important. I'll go over more detail, the DMPK and the adverse event profile and so forth. So what we did is that we evaluated many opportunities from preclinical all the way to phase 3. And basically, we evaluated close to 226 product opportunities, and of that, 152 is in oncology. So we looked at what we did is that we look at those opportunities, and then we evaluated 11 opportunities with the KOLs. And we asked them to say, okay, out of 11 opportunities, what would you recommend? So we talked to the KOLs, we also talked to management, and also our disease biology management to understand what they would like to see, and subsequently, which we licensed in transgene TG4001, which is a therapeutic vaccine for cervical hyperplasia. So looking forward in terms of what did we learn from the past trials and scientifically as well as in the clinical fashion. So we're really looking at cancer cell, which is really heterogeneous, and that's the issue, is that it's constantly mutating. I mean, basically that's the landmark of cancer, right? Mutation, mutation, mutation. And the problem here is that how does the body see it? Does the body see a self-antigen? That nothing to worry about, because cancer cells always send out kind of signals saying, I'm normal, I'm not, I'm not malignant, don't kill me, and it's not. So, but the important thing is that how can we overcome that intolerance? Can we develop some sort of a specific antigen and really kind of enhance that antigen, presenting the dendritic cell, presenting that antigen, and overcome that tolerance and have the cancer cell killed by the immune system? And also understand the immune suppression, because there's always a balance between T effector cells and T suppressor cells, right? So the T effector cell, you want that to go up and suppressor go down, but many times the cancer cells have a way of sending out signals to suppress T regulatory cells. So the question is that how can we overcome that? And of course, we have a therapy now that recently approved by FDA, the ipilimumab, which reduced the CTLA4, which is the T regulatory cells. So with that, then the question here is that what did we learn from the clinical trials? I mean, some of the trials are over again. I see companies coming out with trials. It's typically uncontrolled. And when it's uncontrolled, the question is that, do you have the patient population same as the treatment arm? It's always a question. I also was at a conference that last year around Boston, the FDA. The person's always there is also talk about, if you have a trial, let's do a randomized trial. Although it might be small. I mean, at times, I'm not saying that we only look at late-stage trials, which is really not realistic, because you want to look at innovation. But over and over, I always ask the company the question, what is the target? What is the target? If you have an early trial, that's okay, but show me the target, show me the target modulation, show me the PD marker, that there's a story to tell. So in preclinical study, you show target modulation. In your clinical study, phase 1, you show PD marker, target modulation. You see a story there, and you see efficacy, maybe hints of efficacy. That would do. So, basically, so that's something that we need to keep in mind, is that the PD marker, the mechanism, and how does that complement with existing therapies. So let's talk about the Provenge. So you all know that Provenge is an autologous vaccine, recently got approved, which is really interesting because it shows a more than 4 months difference between the treatment arm or the non-treatment arm and the treatment arm. So that's really interesting that you see that, that the autologous vaccine is making a difference, that the patient's cancer cells, once you take them out, you modify it load it with antigens, and you refuse back, and you see a response there. So that shows that immunotherapy does work. But one thing we have to understand is at the stage of diseases. This is really a non-symptomatic to mildly symptomatic patients. So I always think that when you wanted to treat, in terms of cancer vaccines, you want to treat them early, because once the disease becomes overt, it's really difficult. I mean, you can debulk, but again, it's all mutated, it's all got all kind of cells, and the immune system may be in question. So the important thing is that treat them early. So now I say treatment early, but then you see this immunotherapy with ipilimumab, which is the anti-CTLA-4. Now these patients are pretty sick. Okay, but then still with the immunotherapy, again, this is targeting at T suppressor cells. So cancer cells tend to send out signals, which was suppress the T suppressor cells would reduce. What you try to do is reduce that so that the T effector cell can start working, and by killing the cancer cells. And in this case, you see that patients are pretty advanced in melanoma, and there's a survival benefit. So it's really amazing that you see cancer vaccines working in early setting patients in prostate cancer, and now you see patients with advanced melanoma disease with a couple months to live, and you see this survival benefit. So I talked about the FDA they also understand that the cancer vaccines may warrant different maybe weight of consideration in terms of clinical setting. So they also talked about, this is guidance from 2009, they continue working on it. So again, here they talk about minimal residual disease. So again, you're talking about earlier setting, and the patient population is very important, and how you want to select patient population. You might be able to select patients with certain antigens, the personal health care. So they can select patients, certain antigens, and this way you can actually enhance the clinical benefit that patients are going to be derived from the treatment. Again, PD markers are very important. You can actually monitor the expression and whether or not the patient is deriving a benefit. You can monitor the antigen specific in the CD8 immune response, and we'll see whether or not there's a change in the immune response, particularly responding to that specific antigen. And also monitor that. It's so important. Assay parameters has to be uniform, and statistically analytical methods has been used, because it's so important to make sure that the PD markers is monitored, and you have a validated assay system to monitor that, to show credibility in this way. So, and also important is that there seems to be a delay effect because the body takes time to mount immune response. And you see that with Provenge, you see that with other immunotherapies, about usually 6 months, 8 months delay. So it's important you talk to FDA, because the typical Kaplan-Meier curve may not address that. So let me just kind of go quickly about the prioritization. So important biological response, we're looking at target, mechanism of action. In terms of preclinical, we're looking at magnitude of efficacy. Again, important mechanism, target-driven modulation, and so forth. There's also clinical, I talked about this before, efficacy. Is it the PK is really important, the PD marker again, and development of, and we'll also have to think about development costs and how is that relative to development of pharmaceuticals. So what Roche is looking for is that we're looking for clinical efficacy, but again, we're looking at target modulation, mechanism of action. So in terms of, when we look at opportunities in terms of when it's early, before proof of concept, we're looking for target. Modulation of target, mechanism driven, PD markers. However, once you pass the proof of concept, you have randomized phase two, you show benefit of survival, then it's data driven, but mechanism is very, very important. So I believe that immunotherapy is a great way of treating cancer. I mean, personally, I'm a fan of cancer immunotherapies, and I think I'm very enthusiastic that we might be able to find a way to really make a difference in cancer patients' lives. Thank you.
Any questions? You point out very clearly the immunotherapy, the point is early, treat early, so before the cancer mass become very large. And what do you think of the possibility as a cancer vaccine, even before the cancer cells can be developed? And you already have established immunity to prevent the cancer cell to occur. There are several types of cancers clearly has markers that's known to.
Yeah, good question. When we're doing the therapeutic vaccine task force, we also thought about prophylactic vaccine. And that's a good point because you know that certain markers, COX-2 is a good example, and some others, that may be precursor to developing of cancer. The issue here is the clinical trial. It will be huge. It will be huge. Typically, even in the adjuvant setting, you're talking about huge patient population. So in order to do that, and then also another point is that, before you spend big money on patients, right, you think, well, what's a good preclinical model? And we look at that very hard. Is it monkey? Is it chimpanzees? Is it mouse? We couldn't come up with a good model to really kind of say definitively that this is it. With immune response, the different species have different way of generating immune response. You really need to go to humans. So it's really difficult to say that whether or not, number one, that the investment will be huge before you see any potential difference. And number two is that it's not really a good animal model. You can say that that's going, or this is a good marker for this animal versus humans. Does that answer your question?
One more question.
Based on your 3 criteria for selection, the biological rationale, the preclinical pharmacology, and the clinical data, would your organization consider licensing an oncology vaccine which was not yet in the clinic?
Sorry, say it again last.
Would your organization consider in-licensing an oncology vaccine that is not yet in the clinic?
Yes, that's a good question. The answer will be no, because Roche does not have a vaccine platform. So we really take an opportunistic approach. So like I said, I think minimally we need to see in humans because the immune response is quite different between animal, and then our preclinical folks still does not believe that either monkeys or even mouse will be a good model for it. So at a minimum, we need to see some human data. But I'm not to say that we need to see randomized phase 2. We do look at phase 1, but I look very carefully, our reviewers look very carefully into the mechanism. What is the target? Does it show target modulation? Can you monitor peripherally the markers and say the PD markers saying there's an effect? So if there's an effect, and you kind of see, oh, there's stable diseases, but you see this is decreasing. You have 6 months stable diseases. You can actually see a hint. And once you see the hint, you say, okay, there's something there. Then we will look at it very seriously. So we do look at things early, but then the difference here is that, I guess it's not really, everybody's looking at it, is the mechanism, whether or not it's really a story there. You answer your question?
Thank you very much.
Thank you.